What happens to the body after death? It's a process that's both natural and essential, known as decomposition. Decomposition is the biological breakdown of a once-living organism, and it plays a crucial role in the cycle of life on Earth. Without it, life could not continue as we know it. But what exactly happens during this process? When we think of decomposition, we might imagine a body slowly turning into a skeleton, but it's much more complex than that. It's a process driven by chemical reactions, bacteria, and even insects, all working together to break down the body into simpler forms. In this video, we'll explore the stages of decomposition, the factors that influence how it unfolds, and the science behind it all. While decomposition might seem like a morbid topic, it's actually a fascinating journey into biology, chemistry, and ecology. It reveals how life and death are intricately linked in the natural world, and it helps us understand forensic investigations, environmental ecosystems, and even ancient cultures. So, let's take a closer look at what happens after death and how every part of a living being returns to the earth, continuing the cycle of life. When death occurs, the body begins a remarkable transformation. These immediate changes can be subtle, but they play a vital role in the decomposition process and are crucial in forensic analysis. The first sign is pallor mortis, a noticeable paleness that sets in within 15 to 30 minutes. As the heart stops, blood is no longer circulating, and gravity pulls it down to the lower parts of the body. Without blood flow, the skin in areas like the face and hands becomes pale. This rapid change in appearance is often the first visible indication that death has occurred. Next is rigor mortis, which is the gradual cooling of the body. Immediately after death, the body loses its ability to regulate temperature, and it starts cooling down at a predictable rate, depending on the environment. A body exposed to cold air or water cools faster, while a body in a warm, insulated area cools much more slowly. On average, the body temperature drops by about 1.5 degrees Fahrenheit, or 0.8 degrees Celsius per hour. As the body cools, another process begins: rigor mortis, the stiffening of the muscles. This is caused by the depletion of ADP, the chemical that allows muscles to contract and relax. Without ADP, the muscles lock in place, starting with smaller muscles in the face and gradually extending to the limbs. Rigor mortis peaks about 12 hours after death, and then, after 24 to 48 hours, the muscles begin to soften again as decomposition progresses. During this time, another subtle but important change is happening: livor mortis. As blood settles in the lower parts of the body, it causes purplish discoloration known as lividity. The location and pattern of livor mortis can help investigators determine if a body has been moved after death, making it a critical tool in forensic analysis. These immediate changes, pallor mortis, rigor mortis, rigor mortis, and livor mortis, provide vital clues for scientists and medical examiners in determining the time of death and understanding the body's post-mortem timeline. Understanding these early stages helps set the foundation for the rest of the decomposition process. After death, one of the most well-known physical changes is rigor mortis, the stiffening of muscles throughout the body. This process typically begins a few hours after death, and it marks the third significant change in the body's post-mortem journey. So, what causes rigor mortis? It all starts with a lack of ADP, the energy molecule that muscles need to both contract and relax. When the body is alive, ADP allows muscles to move freely. But after death, ADP production stops and muscles become locked in place, unable to relax. Rigor mortis typically begins within 2 to 6 hours after death, first affecting the smaller muscles like those in the eyelids, jaw, and neck. As time progresses, larger muscles in the arms, legs, and torso begin to stiffen. The process reaches its peak about 12 hours after death, when the entire body is rigid. Forensic investigators can use the timing of rigor mortis to estimate the time of death. However, the environment plays a critical role in this timeline. Cooler temperatures slow down the onset and progression of rigor mortis, while warmer conditions can accelerate the process. Rigor mortis doesn't last forever. About 24 to 48 hours after death, the muscles start to relax as the body moves into the next stage of decomposition. This is because the tissues begin to break down at a cellular level, releasing the muscles from their rigid state. Understanding rigor mortis is key not only in determining the time of death, but also in understanding the complex processes that unfold in the body after death. It's just one part of the fascinating transformation that happens during decomposition. Once the heart stops beating, another important change occurs in the body: livor mortis, or the pooling of blood. With no circulation, blood begins to settle in the lowest parts of the body, pulled by gravity. This phenomenon is known as lividity, and it typically begins within 20 minutes to 3 hours after death. As the blood settles, it creates a purplish-red discoloration on the skin. The areas in contact with the ground or another surface appear pale, as the weight of the body compresses the blood vessels, preventing blood from pooling in those areas. Livor mortis reaches its maximum intensity around 6 to 12 hours after death. By this time, the blood has become fixed, meaning it no longer shifts, even if the body is moved. This is critical for forensic investigators, as lividity patterns can help determine whether the body was moved post-mortem or if it remained in its original position. For example, if a body is found face down, but the livor mortis pattern suggests the blood pooled on the back, it's likely that the body was repositioned after death. Understanding these patterns allows investigators to reconstruct the events surrounding death. Livor mortis, along with other immediate post-mortem changes like rigor mortis, provides vital clues about the timeline of death. By analyzing the progression and distribution of lividity, forensic scientists can estimate when death occurred and whether the body was disturbed afterward. As decomposition progresses, the body begins to break down from the inside through a process known as autolysis or self-digestion. Autolysis is one of the earliest internal changes after death and plays a crucial role in the breakdown of tissues. Autolysis begins in the cells. Once the heart stops beating, oxygen is no longer supplied to the cells, and carbon dioxide begins to build up. This creates an acidic environment within the body, which causes the lysosomes, small structures within the cells, to rupture. Lysosomes contain digestive enzymes, and when they break open, these enzymes are released, starting to break down the cells from within. This process happens first in organs rich in enzymes, such as the liver and pancreas. In fact, the pancreas, due to its high enzyme content, essentially digests itself shortly after death. The soft tissues of these organs begin to liquefy, marking the first stage of internal decomposition. As autolysis continues, tissues break down at a cellular level, creating a nutrient-rich environment that eventually attracts bacteria and other microorganisms, which will further drive the decomposition process. Autolysis sets the stage for the next phase of decomposition, putrefaction. Though autolysis starts internally, its effects soon become visible on the outside of the body. This is the first step in the body's complex transformation from living tissue to the breakdown of organic matter. As decomposition advances, the body enters a stage known as putrefaction, where microorganisms begin to break down tissues on a larger scale. This phase is marked by the release of gases, causing the body to bloat and swell visibly. Putrefaction starts when anaerobic bacteria, organisms that thrive without oxygen, take over the body. These bacteria, primarily found in the intestines, begin to consume the body's tissues, producing gases such as methane, hydrogen sulfide, and ammonia in the process. As these gases build up, they cause the body to inflate, often doubling in size. The gas buildup is most noticeable in the abdomen, but it can also affect the face, limbs, and other areas. This bloat causes skin to stretch and eventually leads to skin slippage, where the outer layers of the skin begin to separate from the underlying tissue. The putrefaction stage is also characterized by the release of strong odors. The foul smell is caused by the breakdown of proteins and fats, releasing chemicals like cadaverine and putrescine. These odors, along with the gas release, attract insects such as flies, which play a key role in further decomposition. The bloat and putrefaction stage is one of the most noticeable phases of decomposition. It continues until the gases are expelled, and the body begins to collapse as tissues further break down. As decomposition moves into the next stage, the body enters active decay. During this phase, the breakdown of tissues accelerates, fluids are released, and the body begins to lose much of its mass. This stage is characterized by rapid decay, driven by microorganisms, insects, and other natural forces. The gases produced during putrefaction are gradually expelled, causing the body to deflate. At the same time, the body's soft tissues, especially the skin and muscles, start to break down. As this happens, fluids seep out of the body and pool around it, further accelerating the decomposition of surrounding tissues. Insects, particularly maggots, play a key role during active decay. These insects feed on the body's tissues, helping to break them down more rapidly. Maggots can consume large amounts of soft tissue in a matter of days, and their activity leaves behind a skeleton that is often still covered in remnants of decayed flesh. During this stage, the body's appearance changes significantly. Soft tissues dissolve, bones become exposed, and the odor from decomposition may start to subside as the majority of gases have been released. The active decay stage is an essential part of the decomposition process, returning nutrients to the soil and facilitating the recycling of organic material. It's the final step before the body enters the skeletonization phase, where only bones remain. After the body passes through active decay, the final stage of decomposition is skeletonization. This stage marks the point where all soft tissues have broken down, and only the bones and some ligaments remain. The process of skeletonization can take weeks, months, or even years, depending on environmental conditions. In warmer, humid climates, skeletonization may occur more rapidly due to the presence of insects and microbial activity. In contrast, in dry or cold environments, skeletonization can be delayed for long periods, even centuries. As the remaining tissues decompose, bones are gradually exposed. Bones themselves don't decay in the same way as soft tissues. Instead, they undergo a slower process of weathering. Over time, bones can break down due to environmental factors such as moisture, temperature fluctuations, and chemical reactions with soil. Skeletonization is often the longest-lasting phase of decomposition. Even though the soft tissues are gone, bones can persist in the environment for years, providing a valuable record of the individual's biological history. Forensic investigators and archaeologists rely heavily on skeletal remains to gather information about age, sex, health, and even cause of death. In the end, skeletonization represents the final stage of the body's return to the earth, completing the cycle of life and death. As bones wither away, the remains of the body fully return to nature, leaving behind only traces of the individual's existence. The process of decomposition is not uniform. It can vary greatly depending on a range of environmental factors that influence how quickly or slowly a body breaks down. Temperature, humidity, exposure to air, water, and the presence of insects all play key roles in determining the speed and nature of decomposition. One of the most significant factors is temperature. In warmer climates, high temperatures accelerate the activity of bacteria, insects, and enzymes, speeding up the breakdown of tissues. Conversely, in colder environments, the process of decomposition slows down significantly. In freezing conditions, decomposition may halt entirely, preserving the body for months or even years. Humidity also plays a critical role. In humid environments, moisture promotes microbial growth and insect activity, hastening decomposition. However, in arid climates, bodies may undergo mummification, where tissues dry out and resist decay, leaving the body partially intact. The presence or absence of insects can make a significant difference in the rate of decomposition. In open environments where insects have access, decomposition tends to proceed more quickly due to the activity of flies, beetles, and other scavengers. However, if a body is sealed in a confined space or buried, it may decompose much more slowly. Decomposition in water is also distinct. Bodies submerged in water decompose differently than those exposed to air, as water slows the drying of tissues and may reduce insect activity. Depending on water temperature and the depth of submersion, decomposition can either accelerate or slow down compared to land environments. These environmental factors, temperature, humidity, insects, and water, are critical in determining how a body decomposes, impacting everything from the timeline of decay to the appearance of remains. For forensic scientists, understanding these variables is essential for determining the time and circumstances of death. Decomposition is a universal process affecting all living organisms after death. However, the rate and stages of decomposition can vary significantly between different species due to a variety of factors such as body size, fat content, and environmental adaptations. For example, larger animals like whales or elephants take much longer to decompose compared to smaller species. This is primarily because larger bodies contain more tissue, which takes more time for bacteria and insects to break down. Their decomposition can last for months or even years in certain environments. On the other hand, smaller animals like rodents or birds decompose much more quickly due to their smaller mass and higher metabolic rates. Their bodies provide less tissue for bacteria and insects to consume, speeding up the entire process. Another key factor in decomposition across species is fat content. Animals with higher fat reserves, such as bears preparing for hibernation, can experience a slower decomposition process. This is because fat acts as an insulator, slowing down bacterial activity and preserving tissues longer than in leaner animals. Decomposition in aquatic species differs as well. Fish and marine mammals decompose more slowly in water due to the cooling effect of water temperatures and reduced insect activity. However, marine scavengers like sharks or crabs can accelerate the breakdown of a body in ocean environments. Insects play a key role across nearly all species, helping to break down tissues. Flies and beetles are often the first to arrive on terrestrial animals, while marine scavengers handle much of the decomposition in ocean species. Decomposition plays a vital role in forensic science, providing investigators with essential clues about the circumstances and time of death. By studying how a body breaks down, forensic experts can reconstruct events leading up to and following death. One of the most important aspects forensic scientists examine is the post-mortem interval, or PMI. This refers to the time that has passed since a person died. By analyzing the different stages of decomposition, pallor mortis, rigor mortis, livor mortis, and putrefaction, scientists can estimate the PMI with remarkable accuracy, especially within the first few days after death. In cases where a body has been decomposing for a longer time, forensic entomology becomes crucial. Insects, particularly flies and beetles, colonize a body in predictable patterns. By studying the types of insects present in their developmental stages, entomologists can estimate how long the body has been exposed to the environment. Forensic scientists also look for signs of taphonomic changes, physical changes to the body caused by environmental factors like weathering, animal scavenging, or soil composition. These changes can help investigators determine whether a body has been moved or tampered with after death. Decomposition can also reveal details about cause of death. For example, bodies that show signs of accelerated decomposition may have been exposed to trauma, toxins, or extreme environmental conditions. By studying the body's decay patterns, forensic pathologists can gather critical information that might not be immediately visible. Through the meticulous study of decomposition, forensic experts are able to piece together the final moments of a person's life, offering valuable insights in criminal investigations and helping to bring justice to victims. While decomposition is a natural process, certain conditions can slow it down or even halt it entirely. This results in preservation or mummification, where the body remains intact for months, years, or even millennia. One form of preservation is natural mummification, which occurs when environmental conditions like extreme dryness, cold, or lack of oxygen prevent the body from decaying. For example, the dry deserts of Egypt created perfect conditions for mummifying bodies naturally. Cold environments, such as glaciers or frozen tundras, can also preserve bodies for thousands of years by halting bacterial activity. In some cases, mummification is artificially induced, as in the case of ancient Egyptian mummification. The process involved removing the internal organs, drying the body with salts, and wrapping it in linen. This meticulous process ensured that the body would be preserved for the afterlife. Mummified remains can offer a wealth of information to archaeologists and scientists, providing a window into the past. By studying the preserved tissues, researchers can learn about the health, diet, and even the causes of death of individuals who lived thousands of years ago. Other forms of preservation include embalming, a modern technique where chemicals like formaldehyde are used to slow down decomposition. Embalming is commonly used in funerals to preserve bodies temporarily, but it can also extend the preservation of remains for scientific or medical purposes. Whether through natural or artificial means, preservation and mummification allow us to glimpse into history, offering insights into ancient civilizations, burial practices, and the biological makeup of our ancestors. Decomposition is not just a process of decay. It's an essential part of the cycle of life and death. Every stage of decomposition plays a role in returning nutrients to the earth, fueling new life and sustaining ecosystems. While the breakdown of a body may seem grim, it's a natural and vital process that ensures life continues. From the initial changes like pallor mortis and rigor mortis to the final stages of skeletonization, every step is part of the body's transformation. Factors like the environment, the presence of insects, and even human intervention through preservation and mummification can shape this journey. Whether rapid or slow, decomposition is inevitable. Forensic science has turned the study of decomposition into a powerful tool for solving crimes and uncovering the mysteries of death. Through careful examination of decay patterns, entomology, and environmental influences, scientists can piece together vital information about time of death, cause of death, and even events that occurred after death. At its core, decomposition is a reminder of the delicate balance of life. It teaches us that death is not an ending, but a transformation. Through the recycling of organic matter, the remains of one life become the building blocks for new growth. In every way, the cycle of life and death is a continuous loop, one where death feeds life, ensuring the survival and renewal of nature.